Welcome back. We are on the second chapter, DNA, RNA, and protein synthesis. The information encoded in genetic material is responsible for establishing and maintaining the cellular and biochemical functions of an organism. In most organisms, the genetic material is a long double-stranded DNA polymer. The sequence of units, deoxyribonucleotides of one DNA strand, is complementary to the deoxyribonucleotides of the other strand. This complementarity enables new DNA molecules to be synthesized with the same linear order of deoxyribonucleotides in each strand as an original DNA molecule. The process of DNA synthesis is called replication. A specific order of deoxyribonucleotide determines the information content of an individual genetic element gene. Some genes encode proteins and others encode only ribonucleic acid RNA molecules. The protein-coding genes, structural genes, are decoded by two successive major cellular processes: RNA synthesis, transcription, and protein synthesis, translation. First, a messenger RNA, mRNA molecule is synthesized from a structural gene using one of the two DNA strands as a template. Second, an individual mRNA molecule interacts with other components including ribosomes, transfer RNAs, tRNAs, and enzymes to produce a protein molecule. A protein consists of a precise sequence of amino acids, which is essential for its activity. Although the deoxyribonucleotide sequences are different in genes encoding different functions and for genes encoding similar functions in different organisms, the chemical compositions are the same. This enables molecular biotechnologists to transfer genes among a variety of organisms to create beneficial products. To understand how this is accomplished, it is helpful to know about the structure of DNA, replication, transcription, and translation. Structure of DNA. The chemistry of DNA has been studied since 1868. By the 1940s, it was known that DNA is made up of individual units called nucleotides that are linked to each other to form long chains. A nucleotide consists of an organic base, a 5-carbon sugar pentose, and a phosphate group. The figure shown here is that of the chemical structure of the components of DNA. A representative a nucleotide, as seen here, deoxyribose, right, adenine, guanine, thymine, and cytosine. Chemical structures of the components of DNA. A representative nucleotide. The term base denotes any of the four bases adenine, guanine, cytosine, and thymine that are found in DNA. The deoxyribose sugar is enclosed by dashed lines. The numbers with primes mark the carbon atoms of the deoxyribose moiety. B. The basis of DNA. The circled nitrogen atom is the size of attachment of the base to the 1' carbon atom of the deoxyribose moiety. Dear candidates, interrupting in between, during the lecture I repeat certain sentences and I am reading every word to the last word in the best interest of those who are, colorblind, or let's say who do not have vision, who are resorting to Braille. I hope my lectures are clear enough to answer your questions. If you do have any concerns, do please mention that in the comment section. This is especially meant for those who cannot read as well, right? Who do not have the gift of vision, but who are resorting to others helping them, or to the Braille language, or to those who are speaking to them. So most welcome to air your thoughts while I read. Thank you. Now continuing with the subject matter proper. The sugar of DNA is 2' deoxyribose because it does not have a hydroxyl group. The 2' carbon, rather, it has a hydroxyl group only on the 3' carbon of the sugar moiety. By contrast, in mRNA, the 5-carbon sugar ribose has hydroxyl groups at both the 2' and 3' carbons of the pentose ring. In both the RNA, the phosphate group and base are attached to the 5' carbon and 1' carbon atoms of the sugar moiety respectively. In DNA, there are 4 kinds of bases: adenine A, guanine G, cytosine C, and thymine T. The nucleotide subunits of DNA are joined by phosphodiester bonds, with the phosphate group of the 5' carbon of one nucleotide linked to the 3' OH group of the deoxyribose of the adjacent nucleotide. A polynucleotide strand has a 3' OH group at one end, the 3' end, and a 5' phosphate group at the other end. Right, the chemical structure of a single strand of a DNA. Right, I'm repeating it. It contains of adenine, a phosphodiester linkage, thymine, another linkage, guanine, another linkage, to cytosine, a 3' and chemical structure of a single strand of DNA. On the left-hand side, we have a rod ribbon model of double helical DNA. The rods represent the complementary base pairs, and the ribbons represent the deoxyribose phosphate backbones. Before we proceed any further, I'd like to express our thanks to the authors of this concerned page or the entire material for the invaluable contribution. It's been a great help to all the candidates who are listening to my lecture. Thank you so much. In 1953, James Watson and Francis Crick used X-ray diffraction analysis of crystallized DNA, discovered that DNA consists of 2 long chain strands that form a double-strand helix. The 2 polynucleotide chains of DNA are held together by hydrogen bonds between the bases of the opposite strands. A base pairing occurs only between specific complementary bases. A pairs only with T, that is, adenine pairs only with thymine, and guanine pairs only with cytosine. So the AT base pairs are held together by 2 hydrogen bonds, and the GC base pairs are held together by 3 hydrogen bonds. The number of complementary base pairs is often used to characterize the length of a double-stranded DNA molecule. For DNA molecules with thousands of millions of base pairs, the designations are kilobase pairs and megabase pairs, respectively. For example, the DNA of human chromosome 1 is one double-stranded helix that has about 263 megabase pairs, right? The AT and GC base pairs lie within the interior of the molecule, and 5' to 3' linked phosphate and deoxyribose components form the backbone of each strand. Figure 2.4. The 2 strands of a duplex DNA molecule run in opposite directions to each other, anti-parallel chains. One chain is oriented in a 3' to 3 or 5' direction, and the other is oriented in a 5' to 3' direction. Because of the base pairing requirements, when one strand of DNA has, for example, the base sequence 5TAGGCAT 3', the complementary strand must be 3' ATCGTA 5'. In this case, the double-stranded form would be, as shown here, this one here. By convention, when DNA is drawn on a horizontal plane, the 5' end of the upper strand is on the left. The genetic material has 2 major functions. It encodes the information for the production of proteins, and it is replicated with a high degree of accuracy to pass the encoded information to new cells. The Watson-Crick model of DNA fully meets these important requirements. First, because of base complementarity, each pre-existing DNA strand can act as a template for the production of a new complementary strand. Figure 24, chemical structure of a double-stranded DNA. You have the adenine, thymine, cytosine, guanine, right? It's on both the sides. So it's about that. So this is the page on the incoming nucleotide with the new hydrogen bonds. We have a DNA replication. The incoming nucleotide is a deoxyribonucleoside triphosphate that is directed by DNA polymerase to pair with the complementary base of the template strand. Consequently, after one round of replication, 2 daughter molecules are produced with each other having the same sequence of nucleotide, and consequently, after one bond of replication, two daughter molecules are reproduced with each having the same sequence of nucleotide base pairs as the original DNA molecule. Second, the sequence of nucleotides of a gene provides the code for the production of a protein. The linear order of amino acids in a protein is determined by the linear sequence of deoxyribonucleotides in a gene. The linear order of amino acids in a protein is determined by the linear sequence of deoxyribonucleotides in a gene. DNA replication. As predicted by the Watson-Crick model of DNA, each strand of an existing DNA molecule acts as a template for the production of a new strand, and the sequence of nucleotides of the synthesized growing strand is determined by base complementarity. During replication, the phosphate group of each incoming nucleotide is enzymatically joined by a phosphodiester linkage to the 3' OH group of the last nucleotide that was incorporated in the growing strand, figure 2.5a, as you can see here. Right, and the nucleotides that are used for DNA replication are triphosphate deoxyribonucleotides that have 3 consecutive phosphate groups attached to the 5' carbon of the deoxyribose sugar moiety. The phosphate that is attached to the 5' carbon is designated that the alpha phosphate. The next phosphate is the beta phosphate, and the third one is the gamma phosphate. During the replication process, the beta and gamma phosphates are cleaved over as a unit, and the alpha phosphate is then linked to the 3' OH group of the previously incorporated nucleotide. The DNA synthesis machinery of prokaryotes and eukaryotes includes a large number of different proteins. Of these, DNA polymerases are responsible for binding deoxyribonucleotides, fitting the correct nucleotides into place according to the base pairing requirement of the template strand and forming the phosphodiester linkage. In bacteria, DNA replication is initiated at a specific region of the usually circular chromosome called the origin of replication, or origin, and in Escherichia coli. In bacteria, DNA replication is initiated at a specific region of the usually circular chromosome called the origin of replication, or origin, and Escherichia coli proceeds at the rate of about 1,000 nucleotides per second. In eukaryotes, a chromosome has many different sites of initiation of replication. Because of these multiple origins of replication, part of the eukaryotic replication process includes enzymatically joining ligating segments of newly synthesized DNA together with phosphodiester bonds. Furthermore, in eukaryotes, a special replication enzyme called telomerase is used for the completion of the linear ends telomerase of each chromosome. Decoding genetic information, Rna&Protein. The vast majority of genes encode information for the production of protein chains. Proteins are essential polymers that are involved in almost all biological functions. They catalyze chemical reactions, transport molecules within cells, escort molecules between cells, control membrane permeability, give support to cells, organs, and body structures, cause movements, provide protection against infections, infectious agents and toxins, and regulate the differential production of other gene products. A protein chain consists of, before going to the next page, let me just complete this a special note, a milestone note, a structure for deoxyribonucleic acid. The elucidation of the structure of the genetic material DNA was undoubtedly one of the most important scientific breakthroughs of the 20th century. James Watson and Francis Crick, who with Morris Wilkins were awarded the Nobel Prize in Physiology or Medicine in 1962 for this work, suggested a structure for the salt of deoxyribonucleic DNA. This structure has novel features which are of considerable biological interest. The structure has 2 helical chains, each coiled around the same axis. Both chains follow right-handed helices, but the sequence of the atoms in the 2 chains ran in opposite directions. The novel feature of the structure is the manner in which the two chains are held together by the purine and pyrimidine bases. They are joined in pairs, a single base from one chain being hydrogen bonded to a single base from the other chain. One of the base pair must be a purine and the other a pyrimidine for bonding to occur. If adenine forms one member of a base pair on either chain, then the other member units must be a thymine. Similarly, for guanine, it would be cytosine. The sequence of base pairs on a single chain does not appear to be restricted in any way. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. In another article a few months later of Nature in 1953, Watson and Crick discussed more implications for their models. The phosphate-sugar backbone of their model is completely regular, but any sequence of the pairs of bases can fit into the structure. It follows that in a long molecule, many different permutations are possible, and it therefore seems likely that the precise sequence for bases is the code which carries the genetic information. That is, that the precise sequence of bases is the code which carries the genetic information. So our model, that is their model, suggests possible explanations for a number of other phenomena. For example, spontaneous mutations may be due to a base occasionally in one of its less likely automated forms. Again, the pairing between homologous chromosomes and meiosis may depend on pairing between specific bases. Within a decade of the demonstration of the double helical nature of DNA with its complementary base pairs, the molecular aspects of DNA replication were known. The cellular processes that are responsible for both decoding and regulating the synthesis of gene products were understood, and many of the kinds of changes that lead to altered gene production were recognized. From the time of its publication to the present, the scientific impact of the Watson-Crick discovery has been pervasive and for the most part inestimable. As one small example, recombinant DNA technology would not exist without knowledge of the structure of DNA. Period. That closes the note there. Now, the structure of a deoxyribonucleotide triphosphate. The phosphate units are designated alpha, beta, and gamma. The alpha phosphate is directly attached to the 5' carbon of the deoxyribose sugar. During DNA synthesis, the alpha phosphate participates in the formation of the phosphate diester bond, and the beta and gamma phosphate groups are released as a unit, pyrophosphate. And continuing with subject matter proper, a specific sequence of units called amino acids is seen. All amino acids have the same basic chemical structure. There is a central carbon atom, the alpha carbon that has a hydrogen, a carboxyl group COO negative, an amino group NH3 positive, and an R group attached to it. Figure 2.7a coming up. An R group can be any one of 20 different side chains groups that make up the 20 different amino acids found in proteins. When R, for example, is a methyl group CH3, then the amino acid is alanine. The amino acids of proteins are designated by either a 3 or a 1-letter notation. For example, alanine is abbreviated ALA or A. In a protein, each amino acid is linked to an adjacent amino acid by a peptide bond that joins the carboxyl group of one amino acid to the amino group of the adjacent one. The first amino acid of a protein has a free amino group N-terminus, and the last amino acid in the polypeptide chain has a free carboxyl group C-terminus. Proteins range in length from about 40 to more than 1,000 amino acid residues. If a protein folds into a particular shape, configuration, or conformation, depending on the location of the specific amino acid residues, and the overall amino acid composition. Individual amino acids have different characteristics that are determined by the properties of their side chains, and these influence the folding of the protein into a particular three-dimensional shape. The shape of a protein in turn helps to determine its function. Also, many functional proteins consist of two or more polypeptide chains. In some cases, multiples of the same polypeptide chains are required for an active protein molecule. Homomeric protein in other instances, a set of different protein chains, subunits, assembles to form a functional protein, heteromeric protein. Finally, large protein complexes that are made up of many different subunits often perform important cellular functions. The decoding of genetic information is carried out through intermediary RNA molecules that are transcribed from discrete regions of the DNA. RNA molecules are linear polynucleotide chains that differ from DNA in two important aspects. First, the sugar moiety of the nucleotides of RNA is ribose, which has hydroxyl groups on both sides of the 2 and 3' carbon atoms, carbons of the sugar. Second, instead of a thymine, the base uracil U is found in RNA. Most RNA molecules are single-stranded, although often there are segments of nucleotides within a single chain that are complementary to each other and form double-stranded regions, infrastrand pairing. The base pairing within a single RNA strand is the same as the base. Before that, the generalized structure of an amino acid and a peptide bond is shown in the figure here. The DNA replication, the incoming nucleotide is a deoxyribonucleotide triphosphate that is directed by a DNA polymerase to pair with the complementary base of the template strand. Consequently, after one round of replication, two daughter molecules are produced, with each having the same sequence of nucleotide pairs as the original DNA molecule. Second, the sequence of nucleotides of a gene provides the code for the production of a protein. The linear order of amino acids in a protein is determined by the linear sequence of deoxyribonucleotides in a gene. DNA replication, as predicted by the Watson-Crick model of DNA, each strand of an existing DNA molecule acts as a template for the production of a new strand, and the sequence of nucleotides of the synthesized growing strand is determined by base complementarity. And during replication, the pairing between complementary sequences of DNA, except that uracil pairs with adenine. Base pairing can occur between two RNA molecules if they contain complementary sequences of base pairs. The major kinds of RNA molecules that are essential for the decoding of genetic information are mRNA, ribosomal RNA, rRNA, and tRNA. The production of RNA from DNA is called transcription. In most prokaryotes, a single RNA polymerase is responsible for the transcription of all types of RNA. In eukaryotic organisms, mRNA, rRNA, and tRNA are each transcribed by a different RNA polymerase. In many of its features, transcription resembles replication. Briefly, one strand of the DNA of a specific region acts as a template for the synthesis of a polymer of ribonucleotides. RNA polymer is sequentially joined where 3', 5' phosphatized linkages, ribonucleotides that are complementary to the nucleotides of the template DNA strand, figure 2.9. As transcription proceeds, the newly synthesized RNA is released from the DNA and the DNA helix reforms. Since only specific segments of DNA molecules are transcribed, sets of short stretches of base pairs with the DNA required to ensure that transcription is initiated at the correct nucleotide and that it terminates at a specific nucleotide. The sequences that control the initiation of transcription usually precede the coding sequence, and the termination signal sequences follow it. The DNA segment that precedes a gene is called the 5' flanking or upstream region, and one following a gene is the is the 3' flanking or downstream region. To initiate transcription, RNA polymerase binds to a specific sequence of nucleotides upstream of the coding sequence known as promoter. Similarly, a specific sequence of nucleotides downstream of the coding sequence known as the transcriptional terminator signals RNA polymerase to stop RNA synthesis. From a molecular perspective, a gene is a specific nucleotide sequence that is transcribed into RNA. Structural genes, which make up the vast majority of transcribed DNA sequences, encode proteins. However, the initial transcription product of a structural gene is an mRNA. In prokaryotes, a contiguous DNA segment forms a structured gene, the coding region. Prokaryotic transcription entails the binding of RNA polymerase to a promoter region. The initiation of transcription at a nucleotide upstream of the structural gene and the cessation of transcription at a termination sequence that lies downstream from the coding region, figure 2.10. In eukaryotic organisms, a structural gene usually consists of several coding regions, exons, that are separated by non-coding regions, introns, or intervening sequences. After RNA polymerase has bound to the promoter and the entire eukaryotic structured gene is transcribed, the introns are removed from the primary transcript, and the exons in the correct order are linked, spliced together to form functional mRNA, figure 2.11 and 2.12. In general, exons tend to be 150 to 300 bases in length, and introns can vary from as few as 40 to over 10,000 bases. A small number of eukaryotic structural genes lack introns. Schematic representation of a prokaryotic structural gene. The promoter region P, the site of initiation and direction of transcription. The right angle arrow and the termination sequence for RNA polymerase T are depicted. A prokaryotic structural gene is transcribed into mRNA and then directly into protein. This is a schematic representation of a eukaryotic structural gene. The promoter region P and the site of initiation and direction of transcription, and the right angled arrow and the termination sequence for RNA. The right angled arrow and the termination sequence for RNA polymerase T are depicted. The number 1 to 5 marks the exons of the structural gene, and the letters A to D mark the introns. The primary transcript is polyadenylated at the 3' end and capped with a modified guanine nucleotide at the 5' end. Processing of the primary transcript removes the introns. The functional RNA is translated into proteins. And in some instances, the introns in a primary transcript may be legitimately removed in more than one way in a process known as alternate splicing. For example, in one kind of tissue, all the exons of the primary transcript may be spliced together to form a functional mRNA, whereas in another tissue, the initial transcript may undergo a different pattern of exon splicing, with an exon being skipped during the process of intron removal and a novel functional mRNA being produced. The exon skipping mechanism generates different gene products in different tissues from the same strand. Before I proceed to the next page, let me just finish up the schematic and the figures out just splicing of a eukaryotic primary RNA transcript. The bracketing arrows mark the sites that are spliced together after the removal of the intervening RNA regions. In this example, introns A and B are spliced out of the primary transcript, and exons 1, 2, and 3 are spliced together to form a functional RNA. Alternative splicing of a eukaryotic primary RNA transcript. The bracketing arrows mark the sites that are spliced together after the removal of the intervening RNA region. In this example, exon 2 flanked by introns A and B is spliced out of the primary transcript, and exons 1 and 3 are spliced together to form a functional mRNA transcript. The structural gene in figure 213, for example, in Drosophila, the fruit fly, that is commonly used in the genetic studies, 2 different mRNAs are produced from the double-sex DSX gene as a consequence of alternate splicing of the exons contained in the gene into figure 2.14. One form is produced exclusively in female flies and the other only in male flies, and each encodes a protein that has a different activity. The protein produced in female fly prevents the development of some male-specific characteristics, including male genitalia, and conversely, the protein produced in male flies prevents the development of female-specific traits. Most, greater than 90% of the RNA in a metabolically active cell is rRNA, found in ribosomes, although there can be hundreds of thousands of different ones. The figure here shows alternative splicing of the double-sex DSX primary transcript produces 2 different mRNAs in the fruit fly, Drosophila melanogaster. Excuse me. The first 3 exons are spliced together in both male and female flies. However, in male flies, exon 4 is skipped, resulting in the splicing of exon 3 to exons 5 and 6. In female flies, exon 3 is spliced to exon 4, which contains a signal for the addition of a poly A, tail PA, resulting in a shortened mRNA. The numbered boxes indicate exons, while the red and blue lines indicate intron sequences that are spliced out of the primary transcript in female and male flies, respectively. mRNAs in a cell, they represent only about 3 to 5% of the cellular RNA, while tRNA represent about 4%. The rRNA combines with the specific proteins to form ribonuclease nucleoprotein complexes that make up the large and small ribosomal subunits. During protein synthesis, one large ribosomal subunit and one small ribosomal subunit combine to form a ribosome. Both the ribosomal subunits of eukaryotes are larger than those of the prokaryotes. This is the amino acid with that anticodon there. Conformation of a charged tRNA. The amino acid is attached to the 3' end of the tRNA, and the location of the anticodon region is enclosed by dashed lines, as you see here. Now, in addition to thousands of ribosomes, a cell that is actively synthesizing proteins has about 60 different types of tRNA molecules. Now, a particular amino acid is enzymatically linked by its carboxyl end to the 3' end of a specific tRNA. For example, the enzyme arginyl tRNA synthetase adds the amino acid arginine to a tRNA molecule. There's at least one tRNA for each of the 20 amino acids found in proteins. After the binding of a particular amino acid to its tRNA, and tRNA is said to be charged in another part of the tRNA molecule, there are three unpaired nucleotides that together are called the anticodon sequence. This sequence plays a crucial role in the formation of the linear array of amino acids that constitutes a protein. The translation part. In prokaryotes which lack a nucleus, the processes of transcription and translation are not spatially separated. When a newly synthesized mRNA molecule begins to emerge from the RNA polymerase complex, the ribosome binds to a specific ribonucleotide sequence near the 5' end of the mRNA to initiate translation. Thus, transcription and translation occur concurrently in a prokaryotic cell. In contrast to eukaryotic cells, a mature mRNA molecule leaves the nucleus by special pores in the nuclear membrane and is bound by ribosomes that either remain in the cytoplasm or associated with the endoplasmic reticulum. In addition to remove the introns before the mRNA leaves the nucleus, it is capped with a modified guanine nucleotide at the 5' end, and a polymer of adenine nucleotides is added to the 3' end to form a poly A tail. The 5' cap and 3' poly A tail aid in the binding of ribosomes to the mRNA to begin translation. Translation requires the interaction of mRNA, charged tRNA, ribosomes, and a large number of protein factors that facilitate the initiation. The transcription figure shows transcription and translation especially separated in eukaryotes. Nuclear pore, endoplasmic reticulum, mRNA cytoplasm. RNA polymerase, primary transcripts, mRNA. Please take screenshots that could help you. Right. Figure 2.19, schematic representation of the initiation of translation in prokaryotes. The mRNA binds to the small ribosomal subunit. For some mRNAs, the shine delgamma sequence near the 5' end of the mRNA base pairs with a sequence near the 3' end of the tRNA of the small ribosomal subunit. The anticodon UAC of the initiator met tRNA base pairs with the start codon AUG of the mRNA. The large ribosomal subunit combines with the initiator tRNA mRNA small ribosomal subunit complex to form the initiation complex. The amino acid methionine of the initiator tRNA is formulated CHO at the amino group in prokaryotes, not depicted. After translation, the initial formal methionine is removed from the protein chain. This is that diagram. Take a note. Back to the subject matter. To the other figure, modification of the ends of a primary RNA transcript in the nucleus. A modified guanine nucleotide cap is added to the 5' end of the transcript, and the polyadenylation signal in the tRNA sequence in the RNA sequence specifies the addition of a polymer of 50 to 250 adenine A nucleotides to the prime end, 3' end to form a poly A tail. The modified ends aid in the transport of the mature mRNA from the nucleus and in the binding of ribosomes to the mRNA and increase the stability of the mRNA. And there we find an elongation and termination of the polypeptide chain in prokaryotes. And translation is initiated by binding of a small ribosomal subunit to an mRNA by base pairing between a sequence of about 8 nucleotides, a Shine-Delgado sequence that is located near the 5' end of the mRNA, and a complementary sequence near the 3' end of the rRNA of the small ribosomal subunit. 3' UAC, 5' anticodon of a specific initiator tRNA. Met tRNA, where F represents F-met tRNA, where F represents a formal moiety that is bound to the methionine residue. It binds to a 5' AUG 3' codon, start codon of the mRNA proteins, initiation factors, facilitate the binding of the initiated tRNA to the mRNA small ribosomal subunit complex. A large ribosomal subunit then combines with the fmet tRNA mRNA small subunit complex to form the initiation complex. In eukaryotes, translation is initiated by the binding of a particular charged initiated tRNA, met along with the initiation factors to a small ribosomal subunit. Next, the 5' cap end of an mRNA, which is combined with the specific proteins, associates with the initiated tRNA small ribosomal subunit complex, and the complex migrates along the mRNA until an AUG sequence initiator or start codon is encountered. The 3' poly A tail of the mRNA facilitates the interactions between the mRNA and the ribosome. When the UAC anticodon sequence of the initiate a met tRNA base pairs with AUG sequence of the mRNA, the migration stops, and the large ribosomal subunit joins the complex to form the initiation complex. The elongation and termination of phases of translation are very similar in prokaryotes and eukaryotes. The elongation process entails the formation of a peptide bond between adjacent amino acids, with the order of the amino acids being determined by the order of codon to the mRNA. More specifically, after the initiation complex is formed, the second set of 3 nucleotides triplet or codon in the mRNA that immediately follows. The AUG codon dictates the anticodon sequence and therefore the charged tRNA that will bind to the ribosome complex. Uncharged tRNAs do not bind efficiently to ribosomes. For example, if the second nucleotide triplet in the mRNA is CUG, the charged tRNA with the anticodon sequence 3' GHC 5' will bind. This charged tRNA carries the amino acid sequence and leucine. This charged tRNA carries the amino acid leucine. Once this charged tRNA is in place and peptide bond is formed, between the carboxyl group the 3' UAC 5' anticodon of a specific initiated tRNA, fmet tRNA, where F represents a formal moiety that is bound to the methionine residue, binds to a 5' AUG 3' codon to start codon of the mRNA protein's initiation factors, facilitate the binding of the initiated tRNA to the mRNA small ribosome subunit complex. A large ribosomal subunit then combines with the fmet tRNA to mRNA small subunit complex to form the initiation complex. In eukaryotes, translation is initiated by the binding of a particular charged initiator tRNA, met tRNA, along with the initiation factors to a small ribosomal subunit. Next, the 5' capped end of an mRNA, which is combined with specific proteins, associates with the initiated tRNA, small ribosomal subunit complex, and the complex migrates along the mRNA until an AUG sequence initiator or start codon is encountered. And the 3' poly A tail of the mRNA facilitates the interaction between the mRNA and the ribosome when the UAC anticodon sequence of the initiator met tRNA base pairs with the AUG sequence of the mRNA. The migration stops and the large ribosomal subunit joins the complex to form the initiation complex. The elongation and termination phases of translation are very similar in prokaryotes and eukaryotes. The elongation process entails the formation of a peptide bond between adjacent amino acids, with the order of the amino acids being determined by the order of codons of the mRNA. More specifically, after the initiation complex is formed, the second set of 3 nucleotides triplet or codon in the mRNA that immediately follows the AUG codon dictates the anticodon sequence and therefore the charged tRNA that will bind to the ribosome complex. Uncharged tRNAs do not bind efficiently to ribosomes, for example. If the second nucleotide triplet in the mRNA is UG, then the charged tRNA with the anticodon sequence and 3' GHC 5' will bind. This charged tRNA carries the amino acid leucine. Once the charged tRNA is in place, the peptide bond is formed between the carboxyl group of the methionine, the amino group of the leucine. The leucine remains bound to its tRNA. Peptide bond formation is catalyzed by activity exclusively associated with the large RNA or RNA. The formation of the peptide bond discharges the initiated tRNA because the bond between the carboxyl group of methionine and its tRNA is cleaved to make the carboxyl group available for peptide bond formation. And the uncharged tRNA is ejected from the ribosomal complex. The methionine-leucine tRNA mRNA combination shifts, translocates along the ribosome to the site vacated by the initiated tRNA, and as a consequence, the next codon of the mRNA is available for binding by another charged tRNA with the appropriate anticodon sequence. If the third codon is UUU, then that's erase, then the charged tRNA with an AAA anticodon will bind. In this case, the tRNA with an AAA anticodon carries the amino acid phenylalanine. Once this charged tRNA is in place, the linkage between the carboxyl group of leucine and its tRNA is broken, and the peptide bond is formed between the carboxyl group of the leucine and the amino group of the phenylalanine. After ejection of the uncharged tRNA, the peptidyl tRNA with the attached methionine-leucine phenylalanine amino acid polymer and mRNA is translocated to a peptidyl site, P site, and the next codon is available for binding by the appropriately charged tRNA in the amino acid site A. The succession of operations that includes binding of a charged tRNA by means of anticodon-codon pairing, peptide bond formation, ejection of an uncharged tRNA, and the translocation continues until all the amino acids that are encoded by the mRNA are linked together. Translation occurs in a 5' 3' direction along the mRNA at a rate of about 15 amino acids per second. When the 5' end of the mRNA is free of a ribosome, it can combine with another initiation complex. A single mRNA can be translated simultaneously by a number of ribosomes, with each ribosome producing a polypeptide chain. In rapidly growing E. coli cells, the entire population of approximately 20,000 ribosomes per cell is capable of producing about 30,000 polypeptides per minute. Parenthetically, the average bacterial structural gene with about 1,000 base pairs, BP, encodes a protein with 333 amino acids because 3 bases code for each amino acid, with the mean molecular weight of an amino acid being about 105. The molecular weight of an average bacterial protein is about 35,000. The elongation process continues until UAA, UAG, or UGA codon, stop codon, or termination codon is encountered. There are no naturally occurring tRNAs with anticodons that are complementary to these codons. However, a protein's termination factor or release factor recognizes a stop codon and binds to the ribosome. After binding of a termination factor, the bond between the last tRNA, which has the complete chain of amino acids linked to it, and its amino acid is broken, resulting in dissociation of the uncharged tRNA, the complete protein, and the mRNA from the ribosome. In addition, a ribosome releasing factor separates the ribosomal subunits so that they can be recycled to the translation of other mRNAs. After translation, a protein may be modified in various ways. In both prokaryotes and eukaryotes, the methionine at the N-terminus is cleaved from most proteins, leaving the second encoded amino acid as the N-terminal moiety. In eukaryotes, certain proteins are selectively cleaved at defined sites processed to make smaller protein chains that have discrete functions. In other instances, especially eukaryotes, phosphate groups, lipids, carbohydrates, or other low molecular weight groups are enzymatically added to certain amino acids of a protein. These chemical additions, post-translation modifications, create proteins that mediate specific cellular activities. The schematic representation of the initiation of translation in eukaryotes, the initiate of tRNA, met tRNA binds to a small ribosomal subunit, and then the mRNA moves along the complex until anticodon UHC of the initiated tRNA base pairs with the start codon AUG of the mRNA. The large ribosomal subunit combines with this mRNA initiator tRNA small ribosomal subunit complex to form the initiator complex. Schematic representation of the elongation. Schematic representation of the elongation phase of translation. The second codon base pairs with the anticodon. The methionine of the initiated tRNA is joined to the leucine tRNA by a peptide bond, and the uncharged initiated tRNA is ejected from the ribosome. Translocation of the peptidyl tRNA and the mRNA to the peptide site from the amino cell site opens the amino cell site for the next codon. The complex genetic code consists of 64 codons. The 3 of these codons are reserved for stops and 1 AUGZ for initiation. When a methionine residue occurs internally in a protein, the codon AUG is recognized by another met tRNA that is neither formulated nor the initiated tRNA. There is one codon UGG for the amino acid tryptophan. For the rest of the amino acids that are found in proteins, there are at least 2, usually 4, and sometimes as many as 6 codons. For example, there are 6 codons UUA, UUG, CW, CUC, CUA, and CUG for the amino acid leucine. Different codons are used to different extents in different organisms. And of the 4 codons for glycine, CGGA is used about 26% of the time by human structural genes and about 9% of the time by protein coding genes of E. coli. The stop codons are also used to different extents in different organisms. In humans, the frequencies of usage of UAA, UAG, and UGA are 0.22, 0.17, and 0.61 respectively, whereas in E. coli they are 0.62, 0.09, and 0.30 respectively. The differences in codon usage, notwithstanding the genetic code, with a few rare exceptions, is the same in all organisms. This is a schematic representation of termination of translation. The stop codon UAG interacts with the termination factor that leads to a termination of translation. The last tRNA is cleaved from the peptide chain and injected. The mRNA and the finished peptide are released. Ribosomes are prepared for recycling by a ribosome releasing factor. So these are the table genetic code and codon usage in E. coli and humans. Just about take a screenshot, it should be helpful. Regulation of mRNA transcription in bacteria. Right? Now, in bacteria, the production of amino acids, nucleotides, and other essential metabolites, replication, transcription, translation, cell growth, catabolic pathways, energy generating systems, and responses to environmental changes, all depend on proteins. However, the energy resources of a cell are not sufficient to support the transcription And translation, expression of all of its structural genes at the same time. Consequently, only those genes that encode proteins that maintain basic cellular functions are expressed continuously. The transcription of the remaining structural genes is regulated. If a protein is required by a cell, then a signaling system initiates transcription, the on state of the pertinent structural genes. Alternatively, if a protein is not needed, transcription of the protein coding gene is turned off into the off state. This lecture ends right now, in a couple of seconds now. We'll be continuing with the remaining portions in the next lecture. So please take screenshot, make notes, be thorough. Best wishes. Okay, thank you.